I welcome today's webinar. Focus on our second enzyme design challenge using our ADMET Design Suite for the in silico design, synthesis, and biological testing of cyclooxygenase COX inhibitors. Michael Lawless, team leader for Chem Informatics Studies, will be presenting today. Joining Michael and myself are Anu Sharma, Senior Marketing and Sales Manager, and Bob Clark, Director of Chem Informatics. An opportunity to ask questions will follow Michael's presentation today. You may either send your question using the questions pane on your control panel, or you may ask your question directly using the hand-raising icon. If you're using a telephone to listen to the call, please be sure to enter the unique audio PIN displayed when you join the call. When you enter your audio PIN, this enables us to unmute your line so that you can be heard. This webinar is being recorded for playback at our website, www.simulations-plus.com. It is now my pleasure to turn the presentation over to Anu, who will be providing a brief introduction to Simulations Plus. Anu?
Thank you, John. Hello, everyone. Before we begin, I would like to give you a brief introduction of our company. Simulations Plus is devoted to developing, modeling, and simulation software. We were founded in 1996, and we are headquartered in Lancaster, California, which is close to Los Angeles. We are a publicly traded company with 34 employees. On the right-hand side, you see is the product portfolio. Our flagship product is GastroPlus, which is used for the simulation and modeling of in vivo absorption, pharmacokinetics, pharmacodynamics, and DDI experiments. ADMET Predictor is another famous product which is used for the estimation of epitox properties of molecules from the structures. MedChem Studio is our chem informatics platform used for data mining and in silico drug design. DDP Plus is used by the formulation chemists in the drug development stage. MedChem Designer is a product for drawing and sketching of chemical structures and can be used by anyone needing to draw molecules. We also offer services, so if you're needing to outsource R&D project, we are available to give you the PV/PK modeling and the chem informatics support as well. So as you can see that our software provides value to the various stages of the R&D program. In the preclinical stage, for the MedChem Studio and the MedChem Designer, these two software provide value in the lead identification and the lead optimization stages. ADMET Predictor is used by a number of scientists in the entire drug discovery phase, such as the DMPK scientists, toxicologists, computational chemists, medicinal chemists, etc. GastroPlus and the DDD Plus, these two software offer value in the later stages of the drug development stage. Before we begin the presentation on the applications of ADMET Predictor and MedChem Studio, we would like to give you a brief introduction on these two software. ADMET Predictor is easy to use, and it rapidly estimates a number of vital assay properties from the molecular structures. You will also be able to build predictive models using your own data. It comes with 5 modules. The physicochemical biopharmaceutical module will allow you to do predictions on important properties such as log P, log T, pKa, solubility, permeability, blood-brain barrier, etc. The metabolism module will allow you to see the metabolites and the sites of metabolism and the CYP enzyme kinetics. Toxicity module will also allow you to measure toxicities on a number of compounds, for example, on HERG, acute rat, phospholipidosis, carcinogenicity, etc. Simulations module will allow you to simulate the fractions of COX and the optimal dose in humans. Our ADMET Modeler, it allows the building of models based on number of methods such as the descriptor, sensitivity analysis, the artificial neural network ensemble, etc. MedChem Studio, it is our data mining software, and it is used for in silico drug design. It comes in 4 modules. The MedChem basic module will allow you to cluster large libraries of compounds, and you will be able to perform a variety of queries, for example, similarity searching, etc. MedChem SAR module will allow you to do the structure-activity relationship analysis and also look at the matched molecular pairs also. The MedChem design module will allow you to do the library enumeration, reaction designing, and to be able to perform functions such as scaffold hopping. The MedChem Studio also comes with the ADMET Predictor module, which will allow you to do ADME-TOX property predictions and also to estimate ADME risk. ADME risk is a proprietary set of rules which you will see more in Mike's talk. We are always devoted to developing our products, so recently we released the ADMET Predictor 7.0, which has several upgrades and enhancements and bug fixes in a number of features and functionalities. So some of the highlights of ADMET Predictor 7.4 is the ionization constant prediction model, which we have significantly enhanced the chemical space coverage. We developed this model in collaboration with Bayer Pharmaceutical. And we also did a webinar on this particular segment, which recording of which is on our website as well. ADMET Predictor is also now compatible with the Linux operating system, and we also have developed an algorithm for computing ADMET risk, so we will be able to estimate the ADME liability in the fuzzy decision boundaries in the molecules as well. Then we have introduced new predictive models. One is the human jejunal permeability model and the PGP flux model, and we have also extended in the toxicity in the environmental space. So we have a model for estimating the likelihood of the chemical biodegradation in environment also. So these are just a few enhancements, but for more you may visit our website. Next is the enhancements in MedChem Studio 4.0. Again, we have one major new feature in that, which is the advanced optical structure recognition feature. So you will be able to extract chemical structure from images in the Word documents, PowerPoint slides, and the web pages. And also there have been minor improvements and performance enhancements in exporting and importing the structures and managing the large data set and the speed of the software in general. So with this, I would like to hand over the presentation to Michael, where you will see the applications of these two software and as the ADMET Design Suite. Thank you.
Thank you, Anu. Good morning, and thanks for joining our webinar. Today I'll be discussing our project to design novel compounds that inhibit cyclooxygenase. In a previous project, we successfully designed antimalarial compounds. However, we're not a pharmaceutical company. We're a software manufacturer, and we want to illustrate how our software can be used in the drug discovery process. So in both these projects, we not only designed the compounds, but we also had them synthesized and tested in biological assays. This next slide is basically an executive summary of the project. So our goal was to develop molecules that inhibit both COX-1 and COX-2 with greater affinity for COX-1 or COX-2 than COX-1. We started with public domain literature of about 550 compounds. Next, we performed computational analysis and design using only the 2D structure of the molecules. So we used our ADMET Design Suite that includes ADMET Predictor for model building and prediction of ADMET properties, MedChem Studio to perform analysis of the compounds and generate new compounds, and MedChem Designer. We also used GastroPlus to perform PBPK simulations on the molecules. We've synthesized 4 molecules so far and tested them. All compounds show activity against both COX-1 and COX-2. The best compound had a COX-1 IC50 of 770 nanomolar and a COX-2 IC50 of 130 nanomolar. This is an outline of the talk. So first I'll discuss the cyclooxygenase background, then discuss our design strategy, the COX data set that comes from literature articles, We built QSAR models from the COX data, and then we also applied QSAR models that are built into ADMET Predictor. The next step was to take known COX-2 inhibitors and lead hop into a new chemistry space. The next step was to select the compounds that we actually wanted synthesized and tested. So this slide discusses prostaglandin production. Prostaglandins act on a number of receptors in an array of different cells. They have a variety of effects. This includes aggregate of platelets. They induce labor, decrease interocular pressure, regulate inflammatory mediation, produce fever, and inhibit acid secretion in the stomach wall. COX kind of sits in the middle of this cascade, and it starts out with diacylglycerol or phosphoglyceride, and these are phospholipids. They're converted to arachidonic acid, which is the substrate for COX, via phospholipase C and phospholipase A2. So here we have the structure of arachidonic acid, so it's a carboxylic acid with some double bonds. Cyclooxygenase actually performs two functions. The first function is to create a cyclic ring, as shown here on the product of cyclooxygenase. When this is created, there's also a peroxide over on this side of the molecule, and the second function of COX is a peroxidase function that converts the peroxide into an OH. So PGE2 is then used as a substrate for prostacyclin synthase to create PGI2. PGE2 synthase and isomerase create another prostaglandin, PGE2, and PGH2 is also a substrate for thromboxane synthase to make thromboxane. There are two forms of cyclooxygenase, COX-1 and COX-2. COX-1 is constitutively expressed in many tissues, and it's the predominant form in gastric mucosa. It's inhibited by anti-inflammatory drugs such as aspirin and ibuprofen. And as I mentioned on the previous slide, prostaglandins are responsible for inhibiting acid secretion in the stomach. So when you take a COX-1 drug like aspirin, it prevents the synthesis of this, which ends up not being able to regulate the acid influx into your stomach, so it can cause gastric ulcerations and stomach problems. In the late 1980s and early 1990s, another form called COX-2 was discovered, and these levels are induced and elevated during inflammation. So the pharmaceutical companies took a hold of this and started developing selective COX-2 inhibitors that were expected to provide relief from inflammation without the gastric problems associated with the COX-1 inhibition. These four compounds were marketed in the late 90s and early 2000s. Starting on the left, we have Vioxx, which is a Merck compound. Celebrex and Bextra were both discovered by GDE Searle, which was then subsequently purchased by Pfizer. And then on the far right is another Merck compound, Turocoxib, and I've also listed the COX-1 and COX-2 IC50s for each one of the compounds and the representative publication of these. As you can see, they're all very active in COX-2 and less active in COX-1. I also show the selectivity of each one of the compounds, and so this is just a ratio of the IC50 in COX-1 to COX-2, and the higher number, the more selective for COX-2. In 2000, Celebrex had sales of over $2 billion. The compound on the right, Turbocoxib, is available in Europe, but has not been approved or even gone through clinical trials, I believe, in the U.S. Vioxx and Bextra, which have the highest selectivity indices for COX-1 and COX-2 were withdrawn from the market due to increased risk of cardiovascular events. So after Vioxx had been withdrawn from the market, there were a number of studies that tried to understand why it was withdrawn and what we can do to prevent it. So Maxwell and Webb in 2005 published an article in The Lancet on COX-2 selective inhibitors, important lessons learned. And one of the lessons or take-home messages they stated was that when they started studying the basic pharmacology of these COX-2 inhibitors, mechanistic studies done shortly after launch indicated clearly that these drugs had the potential to adversely influence the ratio of prostacyclin and thromboxane in the vascular wall. So people were taking these drugs and having a higher risk for stroke. So our hypothesis was that you needed COX-2 selectivity, but that you also needed some COX-1 inhibition so that you would try and maintain this balance a little bit better. This slide is an outline of our overall design and synthesis strategy. So in the first step, we were collected structures and data, and we collected. If you go to Chemble and query for COX-2 inhibitors, you'll get thousands of hits. We only used compounds from Searle and Merck articles to make this more realistic drug discovery project. Now, I guess in the end we could have taken all of the hits, but we just focused on the Merck and GD Searle publications. The next step was to use MedChem Studio to analyze the structure-activity relationships and also molecular pair analysis within the data set. And as you'll see in the next couple slides, there are a lot of very unique things about COX-2 inhibitors, and that you can make a small change and that will result in inactivity. And we needed to understand those when we were designing the molecules so that we designed active compounds rather than inactive compounds. The next step was to use ADMET Predictor to create QSAR models based on the COX-1 and COX-2 data. Afterwards, we took molecules that are known COX-2 inhibitors and lead hopped into new chemistry space using various tools in MedChem Studio. Next, we enumerated the virtual libraries and we predicted their activity and selectivity using the QSAR models that we developed in this step. We also calculated ADMET risk for these compounds, which is an ADMET liability for developing the compounds. And this was performed with ADMET Predictor. We reduced the number of compounds to a reasonably small number and used GastroPlus to perform PBPK simulations on the compounds. So we ended up with a certain number of compounds. We selected to synthesize, and at this point we'd spent about 1.5 FTE months to come all the way up to this stage. Then the final step was to have these compounds synthesized and tested. This slide outlines the data set we used to build the models and perform the lead hopping. We started with about 550 compounds from 20 different journal articles. 480 were from Searle publications and 69 from Merck publications. There were 5 compounds that were in both data sets. I also defined the compounds either as COX-2 or COX-1 inhibitors. The definition here was less than 10 micromolar. So if it was below 10 micromolar in COX-1, or excuse me, in COX-2, but above 10 in COX-1, then it was COX-2 selective. Similarly, if it was more active in COX-1 or less than 10 micromolar in COX-1 and more than 10 micromolar in COX-2, it was COX-1 selective, and then finally if it was active in both, it was neither selective. Overall, the IC50 data ranges for the COX-2 went from 1 nanomolar all the way up to greater than 100 micromolar. The COX-1 IC50s ranged from less than 1 nanomolar, so there's one compound below 1 nanomolar, to greater than 1,000 micromolar. So I just want to go into MedChem Studio now and illustrate how the data was organized. So I got most of the compounds from the original journal article, but I also downloaded structures and data from Kimball and compared them to the data in here and curated the data overall. So as you see, I've read in 549 structures into MedChem Studio. The naming convention contains two parts separated by a hyphen. I took all the journal articles and generated a unique sequential number for each one of those. So this comes from journal reference 8, 7, 8, 18, etc. And then the second part of the name is the compound key within the journal article itself. So this is compound 1AQ from MedChem 1997. I also have a column with a hot link to the journal article. So when I click on that, it brings up the journal article, and then I can scroll down. This one's actually in table 3, and look at the activity of the compound and verify the structure of the compound also. So 1AQ has a COX-2 IC50 of 31 nanomolar, and that's going to correspond to the number in the table. So it was really nice being able to click on this and double check some things, because there's a lot of interesting activity cliffs, and I was always going back and double checking them. I also have a column for a modifier on the IC50 value. And so if it was an exact number, I put an equal there. If it was greater than, I put a greater than. So for example, this compound was tested up to 100 micromolar, and they didn't see any activity at all, so it's greater than 100 micromolar active. I did the same thing for the COX-1 data. I also include a selectivity that's just the ratio of the micromolars. That's this column here, and then this final column, selectivity, is the PIC50 of COX-2 minus the PIC50 of COX-1. The PIC50 column I didn't explain, but it's the negative logarithm of the IC50 when the IC50 is in micromolar units. So a value of 7 would be a 100 nanomolar compound, a value of 8 would be a 10 nanomolar compound, so this is somewhere in between the two. Sort the data on IC50 values, and as you can see, this first one doesn't contain an IC50 value. If you look at the structure over here, it looks like a nice COX-2 inhibitor. In fact, this ring reminds me of Merck's Vioxx compound. So why isn't there an IC50 for this compound? So if we click on the journal article link to bring up the journal article and then scroll down to the second page, lactone 2 was found to be essentially inactive. Only 3 was found to have significant inhibitory activity against COX-2. These compounds are very similar, but you see this carbonyl over here is moved from this side of the ring to this side of the ring, and this is the much more active compound. Just having this carbonyl on the same side as the sulfone group greatly diminishes the activity of the compound. Then finally, just in MedChem Studio, I want to show you how we can quickly find duplicates in the data set. So I simply click OK, and it's selected 5 duplicates, and these are compounds that were tested in both Merck and Searle assays, and they actually got slightly different results that I'll talk about in a few minutes. On this slide, I'd like to point out a few of the activity cliffs within the COX-2 data set. So starting up in the upper left-hand corner, we see this compound with a nitrile over on the substituent along the phenyl rings. If you convert that to a methyl group, you're at 3 nanomolar, whereas the nitrile group is up to 80 micromolar. So it's a pretty inactive compound, but just a very small change. And maybe it's telling us that the pocket that this part of the molecule sits in is limited in size, and this might interfere with that and poke into the side. Then going down into the lower left-hand corner, now we're kind of looking at the other pocket occupied by the phenyl ring, and we see that there's some kind of size constraint potentially in that also, that when the sulfonamide is methylated, we have a COX-2 activity of over 100 micromolar, whereas if we remove that methyl group, we're down to 14 nanomolar, so a very active compound there. So this is the one I showed you in the MedChem studio, so moving the ring, carbonyl from one side of the ring to the other side, results either in greatly inactive or an active compound. Similarly, down in the lower right-hand corner, we see that moving a bromine from the same side as the sulfone to the other side greatly increases the activity here. So you get greater than 30 micromolar going down to 30 nanomolar. Now we see these effects in these two scaffolds, but they weren't transferable to a lot of the different scaffolds. So for example, Bextra has a substituent on the same side of the ring as the sulfone. So I want to go back into MedChem Studio and illustrate generating activity cliffs based on this data. The first thing I need to do is create a single superclass. So I want all the molecules to go in one class, and then I'll use that particular class to generate the activity cliffs. So I click on the Molecule Pairs tab, and up comes a dialog box where I specify the similarity for the molecule pairs and then the difference in activities for the molecule pairs. So I have a criteria here. I'm looking at very small changes, so I want to find compounds that the maximum number of atoms and bonds between the two compounds is less than 4. We'll leave the number sites at 2, and then we'll specify the COX-2 PIC50 for the property to analyze. And if that is 3 or more, then included in this analysis. So I'll click OK, and very quickly MedChem Studio compares every molecule to every other molecule in the data set, generates a maximum common substructure, and then sees what the difference is between those two compounds. If it's less than 4 atoms or mismatched atoms and bonds, then it goes on to the next step where it looks at the COX-2 IC50 data, and if that is greater than 3 in the change, it will present it in this tab. So it's generated 143 of these pairs where the difference is very large as far as the IC50, but the structural change is very small. So this first one is the nitrile going into the methyl group. This next one is a hydroxyl group going to a methoxy group, and that results in a large gain in activity. Similarly, replacing the hydroxyl with chlorine creates a large increase in potency of the compound. So you want to stay away from hydroxyls over on that side as far as this one goes. Another interesting pair here is the removal of the fluorine. So this has 2 fluorine substituents on the phenyl ring. This only has 1. It goes from a 100 micromolar compound down to a 10 nanomolar compound. This pair here where we've got a pyridine being replaced by a benzyl also changes, has a dramatic change in the IC50 value. And this compound over here actually looks a bit like Bextra, or excuse me, urocoxib. Urocoxib would also have another purol. So, you know, why isn't this compound much more active? If we go down to this next pair, here's a pyridine, or excuse me, a pyrazole. So there's 2 nitrogens next to each other in the 5-membered ring. If you convert one of the nitrogens to a carbon to make a pyrrole, you're also looking at a huge increase in activity. So these effects are very subtle, and also you'll note that pyrazole is the scaffold for Celebrex. So if you put a CF3 group in here, this compound also becomes very active. So we not only have to get the right scaffold for the right orientation, there's a lot of electronic effects in there and steric effects that can greatly affect the activity. And then finally, I want to just show that the one with the bromine and the thiophene was also picked up in this analysis. Okay, so on that slide we're showing individual pairs of compounds. Matched molecular pair analysis goes a step further and tries to generate statistics on common transformations or changes. So on this slide we're looking at matched molecular pair analysis for compounds with the methyl sulfone going to a sulfonamide group. So here's a representative example pair. You've got a carbon going to an NH2 group here. The program identified 95 pairs of compounds with this particular structural change. That corresponds to 180 molecules. And we see here the distribution of PIC50 values. Obviously, there's a lot of active compounds in here, but there's some non-active compounds in this region. Then this column looks at the distribution of the change in PIC50 values for these 95 pairs. And the average change is 0.74. So overall, the sulfonamide is more COX-2 active by 0.7 log units than the sulfone. So that's a trend we need to take into consideration when we design these molecules. This next row looks at COX-1. Here we see the average change is about 1 log unit. So sulfonamide here is more COX-2 active by 1.1 log units overall. And then finally we look at the selectivity of the modifications. Here we've defined selectivity as the COX-2 PIC50 minus the COX-1. So the higher the value, the more selective for COX-2. And it's a negative number, and this indicates that the methyl sulfone is more selective than the sulfonamide, and that kind of corresponds to the changes we're seeing up here. Both of these increase by making this modification. Overall, you typically increase the COX-1 and COX-2 activity, and in fact the COX-1 activity by more than the COX-2, so it becomes less selective. So again, let's pop into MedChem Studio. Again, we want to use all the molecules for the pair analysis. We've already got one pair analysis, so the first thing I need to do is remove that. Then when I click on the molecule pairs, again we get the same dialog box. Here I'm only going to specify 2 as the maximum number of atoms and bonds that are not a match, and only have one site. So we're specifically trying to find molecules where the methyl sulfone in this area is converted to a sulfonamide. Again, I'm going to look at the PIC50 for COX-2, but I'm not going to specify any minimum difference. So it'll just find all the pairs there are, and in that way we can analyze everything where this change occurs. So I just click OK, and again, MedChem Studio performs the same comparison. Now, we haven't specified what we want to see in this change, and these first few are just, these are the duplicate molecules. When you get down here, here's the nitrile. We want to be more specific in this area, so we just go down and we do a query. So we do a reaction query, and I've already stored this on disk, but what it is it's a SMRC string that says, okay, find molecules where you have this methyl sulfone that's being converted into a sulfonamide. So I click OK. That selects 95 of these rows, and as you can see, these now have the desired transformation that we want to see. We click on pair clusters. We get several options. One of the options is to create a single cluster using the current selection. So we'll use the 95 and generate the molecular pairs, and then we'll have the distributions for the PIC50s, the chains, the mismatches, etc. And then further down we'll have the values for COX-1 and selectivity. So that's how these tables or rows were generated. And it's very quick in MedChem Studio. Okay, then finally we wanted to look for duplicates, as I illustrated earlier, and so these are the 5 duplicates in the data set, and it shows the Merck value and the IC50 and the Searle value, and then the ratio. And some of these obviously are very close together. Maybe that's kind of an experimental error, or just the uncertainty in the experiment, but some of these, like this bottom one, gets up pretty high at a difference of 6.2. And one reason is they're They made different compounds. We don't know the purity of the compounds. But a main difference is the assays are different. So Merck had two assays. One was performed in microsomes, the other in CHO cells. All the serum data was generated from the soluble enzyme, so they were just looking at an enzyme assay. And since these are enzymes, the IC50 is dependent upon the substrate concentration, so there were slightly different substrate concentrations for each of these assays. We wanted to try and incorporate this data somehow into deriving the models. So we created two descriptor columns, and these are indicator columns to indicate what type of experiment it was. And since there's three different experiments, you only need two indicators. So if the compound was tested for Merck CHO cells, the indicator values were 0 and 1. If it was tested in Merck microsomes, the indicator value was 0 or 1 and 0. And then finally, if it was a Searle assay, it was both zeros. And this was the one that was a larger portion of the data had this. And so we used those as input descriptors in the artificial neural network model for COX-2, and the program selected both these indicators, so they were used in deriving the predictions for the molecules. On this slide, I show the observed COX-2 PIC50 versus the predicted PIC50 from the model. Over on the left is from the training set that contains 384 compounds. Over on the right is the graph for the test compounds, which were 43 compounds. And then the statistics for the regression model here. And you see that we're below 0.05 for the root mean square error on the data. Now, you see I've tested about 400, or I have about 420 molecules in this data set. I couldn't use compounds that had a value of greater than 10 micromolar because we're trying to make a quantitative model, and greater than 10 micromolar is not quantitative, so I had to throw those compounds out for this model. We also created a COX-2 inhibition classification model, and we set the cutoff at 10 micromolar, so if it was less than 10 micromolar, it was COX-2 active. And then I show the confusion matrix for the training set on the left and the test set on the right. So here we have the observed versus predicted values. These are the values compounds we correctly classify with the model, and then across here are the ones that are incorrectly classified. And you'll notice that we get more compounds, or we predict as false negatives than false positives. And I'll tell you why that's important on the next slide. Okay, so we've generated the model, and here are a few of the predictions. So these are some of the Celebrex, Bextra, turbocoxib, and DUP 697. They are all well below 10 micromolar as the IC50, and we correctly predicted that all of these are inhibitors for COX-2. These value in parentheses is the confidence, so we're highly confident in all of these predictions. Over on the right side are some compounds that were more difficult to predict. So here's the compound with the carbonyl on the same side as the sulfonamide. It's not active, and we correctly predicted that as being non-active, but it was a lower confidence. If you move the carbonyl over to here, this is an inhibitor, and we get this value wrong. Now, we can't create perfect models, so that we have to accept that we get a few things wrong, and here we've created a false negative. So we predicted it not to be active, but in reality it was inactive. And I would rather have false positives at this stage, or false negatives at this stage, because if you predict it to be negative, you're not going to synthesize it. So you might miss a compound. On the other hand, if you predict it to be positive, you might go ahead and synthesize the compound, and then it'll be inactive. And we didn't want to make any inactive compounds, so we preferred to have false negatives rather than false positives. And then finally, you'll see we did predict Vioxx correctly, and you'll see that the difference between these two compounds is only this fluorine in this position. So the model was sensitive to very small changes, but you can never get everything right. This next slide shows the COX-1 regression model. Here we have a much more limited number of compounds to predict because remember the literature compounds were always trying to be COX-2 selective, so a lot of the COX-1 activity was greater than 10 micromolar. But again, we've been able to build a reasonably good model here. We also took the ratio of the log of the COX-1 to COX-2 activity and created a selectivity model, and the results are shown on this slide. Okay, so now we've generated a large number of models on the COX data. The next step is to lead hop into novel chemistry space. So one of the first methods we used was combinatorial transformations, and we used Vioxx as the query molecule or the starting molecule. Combinatorial transformations are a set of medicinal chemistry rules for just taking a molecule and modifying it. So for Vioxx, it might have taken something in the ring and converted it to a nitrogen or something like that. So that's one way we generated novel ideas. Another way was to use a procedure called assemble fragments within MedChem Studio. This procedure, the first thing it does is generate fragments, and we used fragments from the World Drug Index, which is and that's a standard file within MedChem Studio. So those are the scaffolds and side chains, and then it would add it to your query molecule, your starting molecule. And here we use several different potent COX-2 inhibitors as a starting compound. We ended up generating about 60 unique molecules, including Vioxx. We eliminated known COX-2 scaffolds, such as the Vioxx one, and then we also removed scaffolds with an sp3 hybridized connection atom. I'll pop back into MedChem Studio again to illustrate these these assembled fragments. So I want to start with the molecules as singletons. So I convert the one group, so everything now is a single compound. I'm going to sort on PIC50 in descending order. So this is one of the most active compounds in the data set. All I need to do is I first need to define the scaffold, and I can do that just by circling the scaffold here. And then I want to go into the reactions and select assemble fragments, which brings up this dialog box. So the input file is automatically included with MedChem Studio, and as I mentioned, it is the fragmentation patterns for the World Drug Index. I need to specify an output file. I'll just put that on the desktop as a CTK file and save that. The operation I'm going to perform is replace the scaffold and retain the R groups. I also specify that the new scaffold must retain the fragment composition. So the R1 and the R2 have to be next to each other in a ring. So click OK, and MedChem Studio generated 41 compounds from this initial starting point. So here's a couple compounds that I would automatically throw out. There's an sp3 hybridized carbon, which is going to make this side chain either come in or out of the plane of the screen, and you need those both in the same plane. So this would be eliminated. Then further down you see some six-membered rings. Let me go ahead and tile this. We see six-membered rings. Here we see the scaffold from aterocoxib. So we've kind of regenerated aterocoxib scaffold. Here's another one that I would throw out. Here's a six-membered heterocyclic ring, some paroles, pyrazoles, isoxazoles, etc. So we have a lot to choose from here. And I think, let's see. Yeah, Vioxx is over here as these two compounds. Okay, so that shows how we generated a lot of novel ideas, and I selected various scaffolds and then put side chains on the scaffold. So each compound was going to contain a phenyl ring with either a methyl sulfone or sulfonamide, and because we didn't know, which side this would work back on we put them on both sides of the scaffold. So in this diagram here, this scaffold represents an unsymmetrical core, whereas these two molecules will be different. And so both those were in the virtual libraries. For the R2 groups, I was pretty conservative on generating those side chains, so I just took molecules that had COX-2 activity of less than 100 nanomolar, and I used those side chains to create the virtual libraries. Overall, we created 9 virtual libraries with 240 compounds each, and then we used our models to predict the COX-1 and COX-2 activity and selectivity. And then we also applied ADMET risk. So ADMET risk I haven't talked about, but it's composed of 3 liability risks, or 3 different types of liability risk. One is absorption risk, so it measures factors for the compound absorbing through the gut membrane and getting into systemic circulation. We also have liabilities for metabolism, so if it's predicted to be metabolized quickly via CYPs, it would get an adverse score. And also we looked at CYP inhibition, which could potentially create molecules that have drug-drug interactions. And then finally we also looked at toxicity, things like hERG liability, etc. This next slide shows the various scaffolds from the virtual libraries and their distribution. So I'm not allowed to disclose any of the structures. So in this first column, this is from the classification model. 0 would indicate it's not a COX-2 inhibitor, and a value of 1 would be the ones that are COX-2 inhibitors. Here's the distribution of COX-2 PIC50s and COX-1 PIC50s. So these look very nice. You're getting very active compounds for COX-2 and then less active predictions for COX-1. Also our ADMET risk, you want values down at the lower end, but you see some going up at the higher. So this one's got more risk than, say, this one, but when we develop these ADMET risk models, you could violate up to 90% of the our subset of the World Drug Index had ADMET risk of 7 or less. So you can still have go up to 7 and still be okay. This next slide shows the remaining 5 scaffolds and again the distributions. Here on these scaffolds, we're seeing a little bit less of, a little bit more compounds are predicted to not be active in COX-2, and in fact this scaffold, scaffold 8, most of the compounds were predicted not to be COX-2 active. And when I looked at this scaffold, it's very hydrophilic, whereas most of the other compounds are more lipophilic. So we started with 2,160 compounds, and then we began filtering them. So with each model, there is applicability domain for the training set. When we applied these to molecules outside the training set, it would ADMET predictor would identify compounds that were outside the scope of the model. So the first thing we did was to eliminate out-of-scope predictions for the COX-2 classification model. That eliminated 52 compounds. Then removed compounds that were predicted not to inhibit COX-2. Then we removed compounds that were out of scope of the 2 regression models. Removed compounds that were predicted to be less than 1 micromolar COX-1. And then if they had too much selectivity, we also throw those out. So we ended up with about 242 compounds. Bob Clark and I went through these compounds and prioritized them. We sent about a smaller set to a committee and then decided to select 8 compounds for synthesis quote. And obviously we want active compounds, so the way we pick these 8 compounds is very important. There were 3 scaffolds, arbitrarily named 1, 2, and 3. Each scaffold had a molecule with the sulfone group on one side or the other. So maybe one was inactive, but the other one should be active. Scaffolds 1 and 2 had 2 ADMET risk codes. They were predicted to have low water solubility and low percent plasma protein unbound, so they were highly plasma protein bound. But again, when you do these predictions on the COX-2 marketed compounds, they also are predicted and in fact experimentally have very low fraction unbound or somewhat insoluble. The synthesis group synthesized 4 out of the 8 compounds, 1 from scaffold 1, 2 from scaffold 2, and 1 from scaffold 3. So in scaffold 2, we're able to experimentally verify what's more potent with the sulfone group, if that's on the same site as the substituent or not. This next slide shows the experimental results for the COX-1 and COX-2 IC50s. So the first compound from scaffold 1 is just weakly active, 23 micromolar in COX-1 and 11 micromolar in COX-2. These next 2 compounds are of the same core, and we have the same substituents, but we just switch the substituents around. The first compound has the sulfone on the same side as the substituent, and that shows a preference for COX-1 over COX-2. This next compound switches it to the other side, and you see we get a large boost in COX-2 activity here, but it also increases the COX-1 activity, so the selectivity is not quite what we want. And then our final compound, which we made one of these with this particular scaffold, ended up being our best compound, 770 nanomolar in COX-1 and 13 nanomolar in COX-2 with the selectivity of 6. So I'm very pleased with these results. We're taking data from somebody else's assay, designing compounds, and then the assay that Sarep performed was actually a 4th variation on the assay, so it wasn't exactly similar to the Merck or the Searle. Here I show the dose response curves for the SLP20 compound, and this is against a percent control, but you'll see down at the bottom that we are achieving 100% inhibition relative to the control, and that this bottom part of the curve is nicely flat and formed. This next slide shows the predicted solubilities of the compounds and log P, and we get all these predictions very nice. It's kind of interesting that these two are flip-flop from the predictions in their experimental values. The log P predictions are all quite nice here. So we also had absorption systems run some assays where they were looking at plasma protein binding and whole blood to plasma ratio. And they got some kind of odd numbers from that. So they did a study of plasma stability. And so they put the compound in plasma and then waited 2 hours and saw what percent remained. And so this first compound was pretty unstable in plasma, which is 5%. The next 2 compounds, the next compound's more stable, but still under 50%, and then these last 2 compounds are reasonably stable in plasma protein. And we're trying to get a handle on what's going on here. We think there's a particular enzyme in the plasma that's responsible for this, but there is some SAR involved in this because these are somewhat similar compounds. And then finally, let me go into this last column because I'm running out of a little bit of time. Here's the experimental human liver microsome clearance. The compounds were somewhat stable in human liver microsomes. This last compound has a fairly high clearance, so it was cleared fairly quickly, and we can look at our predictions from MedChem, from ADMET Predictor, and there's the R2 substituent here. I think we can make quite a bit more stable, and I think that's why this has a fairly high clearance. So then on this final slide, I just want to summarize the project. Every molecule that was designed and synthesized is an inhibitor of both COX-1 and COX-2. And I think this is an excellent starting point for additional optimization of the compounds. And I think we can tune these compounds to have various selectivities. So very positive about the results. And then finally, I'd just like to thank or acknowledge my co-workers, Bob Clark, who's the Director of Chemoinformatics, Mike Bolger is our Chief Scientist at Simulations Plus, and then Walt Wotas is our Chief Executive Officer. The assays were performed at CEREP and Absorption Systems. CEREP did the COX-1, COX-2 assays as well as the solubility. Absorption System performed the HLM studies and some of the other values in there. And then Southern Research Institute synthesized the compounds. And I thank you for your time today, and I know we're running a little bit short of time, so if you have any other questions we're not able to get to, you can certainly email us. Thank you.
Michael, thank you so much for the excellent presentation. We do have a couple of minutes left to address a few questions. Once again, you can either write in your questions on the little chat box, or if you'd like to ask a question using your telephone, please use the hand-raising icon on your control panel and be sure to enter your unique audio PIN. I believe there's one or two questions that have come in written, and Bob, I believe you'll address the first one.
It's a question from Leela Mashwari asking, does the arachidonic acid produce the same that is involved in cocoons during weaving? I'm not sure which acid Leela is asking about. I think it's what she put in is a misspelling, and it may come from the fact that it prompted me to do some web work, and I actually learned something very useful, which is arachidonic acid, despite its name, has nothing to do with spiders, except that it's the involved in the response of your body if you get bitten by a spider. The ARACH part of arachidonic acid comes from the Latin for legume, and that's because you get the arachidonic acid and arachidic acid from peanut oil. The spider cocoons, like caterpillar cocoons and the spider silk that the spiders make webs from, are primarily proteins. So they're composed of amino acids, which are really structurally quite different than the arachidonic acid. I hope that addresses Leela's question.
Yeah, thank you so much, Bob. There's another question that came in. How many heteroatoms are in the central scaffold of the tested compounds? Is this something that might be related with the mispredictions for CYP stability?
Bob, you want to try that one? I don't know. We don't want to disclose structures.
The answer, I believe, is no. That probably does not have anything to do with the errors in the CYP stability. Although the errors in the CYP stability in the slide Michael put up are actually quite small. I mean, they're mostly within a factor of less than 2, and the worst is a factor of 3. And the errors on our individual models, which we're adding up here, are about 0.5 log units, which is a factor of 3. So in fact, it performed pretty much as expected on these predictions. And I should add, these predictions are very hard. I believe they were all in scope, weren't they, Michael?
Yes, I think so.
Because we have internal flags that warn us if we have a structure whose properties or molecular properties are too far away from our training sets.
Thank you, Bob. Thank you, Michael.
You're welcome.
Another question has just come in. How do we make sure the perfect balance between COX-1 and COX-2 inhibition is maintained to design better inhibitors? The $64,000 question.
That is a good question. I mean, it probably, you know, would be answered in the clinic by testing compounds with different IC50 values for these in ratios. So it's a difficult question. I think you'd have to do some clinical trials, and I think the advantage of the compounds we've designed here is that I think we can modify these and tweak the ratio of COX-1 and COX-2 activities to get either COX-1 selective or COX-2 selective, and then kind of increments in between there. But it is a difficult question to answer, and I don't think you can answer it a priori. I think it will need some trials.
I want to do a quick follow-up on Bronco's question about the scaffold stability.
Mm-hmm.
And that is the big instability that we saw, which we had not anticipated. We think we know what enzyme it's coming from, but it really is completely out of left field. It's not something we have any models for, or that we had any particular reason to expect to be attacking our compounds.
Okay, thank you, Bob. Thank you, Michael.
Well, you're welcome.
Another question, have the synthesized compounds been tested for hERG inhibition?
No, we didn't test them in hERG. hERG has a particular pharmacophore, and we didn't really have that pharmacophore really close in these molecules, and so all the predictions we performed on it said that it wouldn't bind and inhibit hERG. So, that is a test, though, that I guess, if you wanted for regulatory committees, they probably would want some kind of hERG test run on those, but we haven't run those.
Thank you, Michael.
Mm-hmm.
I'm going to do my best here with this one. Can more thiophene or chlorothiophene be studied? Does that ring a bell?
Yeah, I guess one of the scaffolds had a bromochlorophene. So yeah, that might be one of your starting structures to modify and go from there. So here's an example down here. So in fact, this molecule here, this DUP697, was kind of one of the starting points for Merck and 2D Searle, I believe. So I think this came maybe from a screening hit or something like that. Now, I'll let Bob give his opinion on thiophene as drug molecules.
A lot of companies avoid thiophenes. Bromothiophene, halothiophene like this at the alpha position tend to be pretty reactive. For example, with glutathione. And if that position is not substituted, they tend to get oxidized pretty rapidly in the biological system, mostly by CYPs. And when they do, they have a nasty habit of causing CYP inhibition, irreversible CYP inhibition and liver damage. So presumably that's why that lead was not followed up. It's very active and it's pretty selective.
Thank you both. Another question, how much correlation with in vitro IC50 values in vivo models like mouse paw model?
Yeah, that would be the next step is to try and run these in some kind of mouse paw model for arthritis. That's one thing I'd like to look at the literature compounds and try and compare their in vitro IC50 values to the in vivo experiments. So that is a good question, and it'd be investigated by looking more in depth at the Searle and Merck papers and trying to get this correlation between the two. So that's a good question, but we haven't done any of that testing yet.
Yeah, the correlation for COX-1 especially is not that great, partly because there are several COX-1 inhibitors, especially ones that are carboxylic acids, which undergo a slow, tight binding interaction so that the usual in vitro assay doesn't pick it up.
Interesting. Okay.
Okay, thank you both. One last one that just came in. How can this software be applied to test for natural product inhibitors of cancer?
I guess you could run the natural products through our model. So COX-2, actually one of the reasons Vioxx ended up being thrown out is they started doing clinical trials at larger doses to look at colon cancer and how COX-2 might be able to attack that. So that's kind of the connection between COX-2 and cancer. So if you had natural products, we could screen them through the COX-2 assay or the model to see if any of these showed activity. Now they probably have to be somewhat similar to the model. They'd have to be in an applicability domain of the models for these to go through. Another avenue or a recent publication I saw was that a group had taken COX-2 inhibitors and run them through a cancer screen and then optimized the compounds for the cancer screen. So they kind of went from a COX-2 inhibitor into an anti-cancer compound. I think that was published in the European Journal of Medicinal Chemistry.
The other point to make here is that sometimes people think that because it's called ADMET Predictor, and many of the models are focused on human responses or human pharmacokinetic properties, that it's only trained on drugs, and that's not true. Many of the models, especially toxicity models, but also the physical property models and the metabolic models, include a lot of other compounds, including some natural products, and very non-drug-like compounds. So the fact that something is a natural product doesn't necessarily mean that the model it'll be outside the applicability domain. That's determined by the molecular properties of the molecules in the training set, and the training set is much broader than just drug-like molecules.
Okay, thank you, Bob. Thank you, Michael. Okay, as you can see from this slide here, we will be at a couple of conferences coming up over the next 2 months or so. Please feel free to stop by and visit with us to learn more about the products. And then, Michael, if you go to the last one, it just shows some contact details. Should there be any questions about the software products themselves or the consulting services that we offer, please feel free to contact any one of us. That's going to conclude today's webinar. If you missed any part of today's presentation, the recording is going to be available within the next day or two at our website, www.simulations-plus.com. Thank you so much for joining us, and have a wonderful day. Bye-bye.